Good day, everyone, and I'm happy to present an update on immunomodulatory therapy for chronic hepatitis B from the AASLD Digital Liver Meeting. So these are my disclosures, and I'd like to start off with a brief rationale for immunological targets. So to date, elimination of HBV infection has only been achieved by the immune system, and a successful immune response has been well characterized in the resolution of acute HBV infection. In terms of innate immunity, we know specific cytokines like interferon alpha, IL-12, and IL-18 can have an antiviral effect. For adaptive immunity, it's a coordinated, robust CD4 and CD8 T cell response and B cell response. The CD4 T cells are required for an effective CD8 T cell differentiation. These CD8 T cells then produce interferon gamma, which non-cytolytically leads to HBV reduction in HBV replication. And CD8 T cells also eliminate infected hepatocytes, while the antibodies produced by B cells clear circulating virus and antigen. So the objective of immunotherapy for chronic hepatitis B has been to restore the level of immune function observed during resolution of acute infection. But are the requirements for HBV control the same during chronic infection? So we know the starting point to stimulate HBV immunity is vastly different in chronic hepatitis B patients. The innate immune system changes dynamically with the inflammatory environment, and this has likely led to some of the confusion observed in the literature. The literature has clearly shown that recruitment and differentiation of myeloid cells during liver inflammation, in addition to suppression of innate lymphocyte function. And this is really a consequence of the heterogeneous patient population. In terms of the adaptive immune response, it has been well studied and well characterized that there's a profound exhaustion of HBV-specific T cells. This results in a narrowing of the T cell receptor repertoire, as well as impaired antiviral function. And recent data also indicates that B cells are also sensitive to this exhaustion, resulting in impaired anti-HBs antibody production. So I want to jump right in then and start with innate immunomodulation. So there was only one study presented at AASLD looking at innate immunomodulation, and this was the TLR8 agonist from Gilead, selgantolimod. This was a phase 2 randomized double-blind placebo-controlled study where patients were viremic upon enrollment and then started either tenofovir alafenamide alone or in combination with selgantolimod at 1.5 and 3 milligrams per kilogram. So I'm not going to show the patient characteristics, but I do want to cover the ALT elevations because this is a primary concern with any immunotherapy. And so ultimately, there was an equal, essentially 15% of patients in each group experienced ALT elevations. They were slightly higher with selgantolimod combinations in both groups, between 2.5 and 5 times upper limit of normal. There was one patient with greater than 5 times upper limit normal ALT elevation, but this patient was enrolled with the 350 ALTs, an ALT of 350. So looking at HBs antigen reduction after selgantolimod treatment, you can see that either the 1.5 or the 3 milligram dose of selgantolimod did lead to some HBs antigen reduction, but this was not dramatically more than the placebo alone. And this data was somewhat mirrored in the HBV DNA levels, where the combination therapy was not significantly different from that observed with tenofovir alafenamide alone. So there was another poster presented by Gilead to demonstrate that these drugs are in fact working in chronic hepatitis B patients. And in this study, they compared both the patients that were virally suppressed at enrollment and those that were viremic and looked at cytokines in the peripheral blood. So here, this is the 1.5 milligram is in green and the 3 milligram is in blue. And what you can see is that at each dose, the patients are responding, and this response does not decrease with time, and it's not different between the virally suppressed and the viremic patients. And this is true for IL-12, IL-1RA, as well as the antiviral cytokine interferon gamma. So, we have to start to understand where the disconnect is in this translation, because now, we've seen 2 drugs come through phase 3 trials, one targeting TLR7, which induces interferon alpha, and another one now targeting TLR8, which induces interferon gamma. Both have shown good efficacy in animal models, and both have been validated in terms of target engagement with their immune biomarkers measured in the periphery. So interferon stimulated genes for TLR7, and as I just showed you, IL-12 and interferon gamma for TLR8. So I think there's some important questions that we have to address here is one, how much of this is actually getting to the liver, because these are orally delivered drugs. They're going through the intestinal tract, through the portal vein, where they can finally get to the liver to engage either dendritic cells or monocytes and macrophages. And I think it's going to be important to understand how much of that effect is observed in the liver versus the gut. And then a second question that hasn't really been investigated is, really, does non-cytolytic HBV clearance play a smaller role in humans compared to the animal models where these drugs have shown very good efficacy? So then, I want to move on to modulating the T-cell response, and in this case, looking at therapeutic vaccination. And there was one study presented at ASLD looking at 18-month follow-up to vaccination, and this is the NASVAC study. This is a vaccine that's comprised of recombinant HBs and HB core antigens delivered intranasally as 10 doses every 2 weeks. And they tested patients that were on nucleoside analog treatment and patients that were not on treatment. And so this is the data in HBs decline. On the left are patients that were on nuke treatment and the patients without nuke treatment on the right. And overall, the HBs decline was relatively modest at the 18-week follow-up with 25% or 27% decline. 2 patients in the nuke treated group had lost HBs antigen, whereas 4 patients that were not on nuke treatment had lost S antigen by the end of the 18-week follow-up. They also looked at anti-HBs in the serum of these patients and found induction of anti-HBs in both groups. The nuke-treated groups started with 3% of the patients at enrollment being anti-HBs positive, with 31% of those patients positive at the end. Whereas in the patients without nucleoside analog treatment, 21% were positive at enrollment. And then after 18-month follow-up, 57% had anti-HBs antibodies in their serum. So, overall, the HBs decline was quite low in this group. There was no placebo arm to compare to, and it wasn't clear if the patients that lost HBs antigen were positive at enrollment or became anti-HBs during the vaccination. So there was another therapeutic vaccine presented as a poster from Virion Therapeutics. This is a preclinical vaccine comprised of the N-terminus of the HBV polymerase. It's fused to a herpes simplex virus, glycoprotein D. They deliver this vaccine as an adenoviral vector, and the glycoprotein D, in this case, acts as a checkpoint inhibitor. And they tested immunogenicity in the AAV 1.3 HBV mouse model. And so the blue here is in naive mice, showing T-cell induction in naive mice. The red is showing T-cell induction in the HBV mice. And this induction of T-cell immunity in the HBV model was showed reduction of viral load in the AAV HBV mouse. They also demonstrated the induction of polymerase-specific CD8 T-cells in the liver of HBV mice. Further studies looked at the diversity of response to the vaccination. And you can see the diversity here, the breadth of the response in the naive mice compared to the low viral load AAV HBV mice showed a similar amount of diversity in terms of vaccine-induced T cells. Whereas if the amount of AAV HBV was increased, this T cell response started to be blunted, but was still to some extent diverse. So some of the caveats of this is that there was no control for the glycoprotein D to understand its checkpoint inhibitor effect. And then I would also say to some extent the effectiveness of polymerase-specific CD8 T cells remains unclear in humans. So then moving on to checkpoint inhibitors. So we all know that there's no shortage of suppressive factors in the liver, but it's really this PD-1, PD-L1 interaction that's being targeted as therapy and cautiously moving towards treatment for chronic hepatitis B. And we're also all aware of this pioneering study from Gilead and Ed Gehring looking at nivolumab in chronic hepatitis B patients. And this one patient now has been well characterized that lost HBs antigen during this study. And some of the earlier data that was presented regarding this patient looked at the magnitude of this T cell response. And this patient that lost S antigen was presented here in red dots. And there was nothing remarkable about the magnitude of the T cell response in this patient. And I would say that's where this heterogeneity of response and not understanding what biomarkers lead to this HBs antigen reduction in PD-1 blocking has led to a lot of translational research that was presented at ASLD trying to improve patient selection. And so this is a study from the group of Carlo Ferrari looking at how the level of exhaustion relates to checkpoint inhibitor therapy. They use flow cytometry data to compare CD127 and PD-1 expression on core, HBV core specific CD8 T cells to establish an exhaustion index. And so low exhaustion index are green and blue, indicated by high CD127 expression, and highly exhausted T cells are in red, indicated by high PD-1 and low CD127. And so as controls in this study, they used patients that had achieved functional cure, either spontaneously or on new treatment. And you can see a majority of the HBV core specific CD8 T cells show a low exhaustion index. And then in HBs positive in chronic hepatitis B patients with HBs antigen, they found that there was two groups of patients here, one with a low exhaustion index in the gray highlight, and then patients with a high exhaustion index. And when they compared this exhaustion index to the functional profile of of these cells, you can see that as the exhaustion index goes down, the functional profile goes up. And when they tested these low exhaustion index patients with PD-L1 blocking with an antibody or small molecule, these patients with low exhaustion index showed the best response to anti-PD-1 blocking. So this suggests that phenotypic markers may improve patient selection. So HBV-specific CD8 T cell analysis in this really presents a challenge ex vivo, but it may allow for the potential to generalize biomarkers for patient selection. So there was also a follow-up study from Gilead looking at these patients that had a significant S decline during the nivolumab treatment. Here, they looked at 3 time points along the course of treatment. So baseline, week 12 and week 24 in 4 patients. So 2 that had HBs reduction and 2 that did not respond to nivolumab treatment. And they performed single cell RNA sequencing on these patients and clustered the T cells and then looked for the distribution of the prevalent T cell clones using the TCR sequencing approach. And seen here in this top row is responder 1, looking at the baseline week 12 and week 24 time point. And you can see that the first 2 time points, the prevalent T cell clones are overlapping, but at this 3rd time point, there seems to be an expansion of unique T cell clones. And when you look across the other responder or in the non-responders, you don't see this change in the profile or the clones of T cells. So, you know, at this point, it's difficult to understand what this means. It's a N of 1. This could be an expansion of subdominant clones, but it may indicate that there is unique T cell expansion in response to PD-L1 or PD-1 blocking. And then lastly, on the checkpoint inhibitors, there was a study to look at trying to improve the timing of delivery in combination with therapeutic vaccines. So this was from Janssen Pharmaceutical or Janssen Infectious Disease. I presented this poster using therapeutic vaccination in the AAV HBV mouse model. And you can see vaccination was delivered at day 28 and day 49, and each of the colored dots are the timing of checkpoint blockade in relation to the therapeutic vaccine. And then they looked at T cell expansion in these mice, in the spleen, and in the liver. And in both cases, the therapeutic vaccine alone is group 1. And overall, you can see that the blockade of response appeared better in the liver compared to the spleen. And the investigators really suggest that blocking after the boost is best. So later, after the second boost of the therapeutic vaccine. But I would say overall, the impact is quite small. And one important point was that blockade prior to vaccination was not actually tested in this study. And the last thing I want to talk about is a novel T cell receptor-based therapy that was presented by Immunocore at ASLD. And these are MTAV molecules. So MTAV is immune mobilizing monoclonal T cell receptors against the virus. And these are currently in phase 1 for chronic hepatitis B. These are soluble bifunctional T cell receptors. And these MTAV molecules differ from a natural T cell receptor because they're, one, not membrane bound. They do not associate with the CD3 and instead have an effector domain that actually engages CD3 on polyclonal T cells. So these receptors remain specific for the MHC peptide presentation and engage polyclonal T cells by binding CD3 on the T cells. And so they recently published the preclinical data for this and show that, in fact, you can target the intracellular antigens using this approach, and that these molecules are specific for HBV infected and integrated cells, and they are not impacted by inhibitory receptor expression because they're using a polyclonal T cell population and not HBV specific T cells. And so this is just one piece of data from the recently published paper, and this is the HepG2 NTCP in vitro infection model. And so when HepG2s are infected with HBV, you see viral replication. When you just add the T cells to this co-culture, there's no impact on viral replication. But when the MTAV molecules are titrated in, there's now a significant reduction in HBV replication because you've now activated the polyclonal T cells to the HBV infected HepG2 cells. So one limitation, of course, of T cell receptor based therapies is always HLA restriction, and an interesting concept in ImmunoCore also presented during the meeting was the use of HLA-E, which is non-classical and expressed throughout the population. So that could be a potential way to generalize T cell receptor therapy in the chronic hepatitis B patient population. So with that, I would just like to finish and give a brief summary and perspective. So the therapeutic vaccines, so it's clear that major obstacles still remain for immunogenicity in chronic hepatitis B patients. It's unlikely to be successful as a monotherapy, and it's likely going to require combination. And there's interesting strategies that are becoming available, such as antigen reduction, combination with checkpoint inhibitors, and I think also a potential role for innate immunomodulation in the liver. In terms of innate immunomodulators, so despite inducing the cytokines with known antiviral function, they have not been effective as monotherapy. I say a good thing is that they have demonstrated safety, and so I think this does justify combination with additional immune targeting drugs where you can make the liver environment amenable to an immune response. And then in terms of checkpoint inhibitors, these remain promising, but patient heterogeneity is a significant issue. And you can see from the translational research that was presented during ASLD, defining the biomarkers to select these patients is likely to improve the safety and efficacy of this class of drugs. So thank you very much, and I'm looking forward to the discussion.